Hey, got a recording. Okay. Yeah, so today's seminar, we have a very recent graduate from the Wallace Lab. We have Sebastian Haas, who's going to talk to us a little bit about what looks like the Bedford Basin there. So some local stuff, more than that. Yeah, take it away.
Thanks, Dave. So today I'll be talking about, welcome everybody, first of all, and thanks for attending. Today I'll be talking about a bunch of stuff I've done over the past 5+ years as a PhD student in the Wallace Lab here at Dalhousie's Oceanography department. And if you've been to my fairly recent defense, I think you'll still find this interesting because I try to mix it up a little bit and add more emphasis on the future work that can come out of the thesis and look at some of the stuff I presented at the defense also from a different angle. So I titled my talk Microbial Nitrogen Cycling Along Space and Time, the Biogeochemistry of Two Coastal Water Columns with Contrasting Mixing Regimes. And of course, I have to start by introducing the aquatic nitrogen cycle, which I'm sure everybody loves as much as I do in all of its complexity. But I try to focus on some of the core processes that are very important for the nitrogen cycle itself and also for the results I'm going to present today. Starting with a process of nitrogen fixation, which makes the abundant atmospheric N2 bioavailable to the vast majority of organisms that do not have the ability to fix nitrogen, N2. And then there's nitrification, which uses the ammonium produced by nitrogen fixes to oxidize it via nitrite to nitrate. And again, you need specialized microbes to do that, and you need oxygen. And the last process I want to highlight here is denitrification, which is kind of the flip side of N2 fixation and removes bioavailable nitrogen, again, out of reach from the majority of organisms. And here you need require an absence of oxygen for denitrification to occur. And to get back to nitrification, nitrification is a prerequisite for nitrogen removal by denitrification, which cannot occur from ammonium. It needs nitrite or nitrate to proceed. And so that's one importance of nitrification. Another one, slightly less appreciated generally, is its ability to determine the type of nitrogen in the environment and available to primary producers. And we know from literature that the ratio of ammonium to nitrate in the environment can have a big impact on the ecology of phytoplankton, its community composition. And for example, high ratios of ammonium to nitrate, so high ammonium, has been associated with harmful algae blooms and the phytoplankton type that's associated with those. A little more on nitrification. So it's this two-step process where the first step from ammonium to nitrite is catalyzed by ammonia oxidizing bacteria, which I'll be abbreviating AOB, or ammonia oxidizing archaea, AOA. And in my research, I use functional marker genes, which are so you can go into the environment, sequence the DNA from a sample, and look at the distribution over samples of functional marker genes. And what they are is they can give you an idea of the functional potential for a given process in the environment. So, for example, MOA is the gene that codes for the enzyme that catalyzes part of the reduction from ammonium oxidation from reaction from ammonium to nitrite. Another technique I use in my research is stable isotopes of nitrogen, and for this we need these quantities of enrichment factors, isotopic enrichment factors. Here the 15 epsilon AO is the isotopic enrichment factor for ammonia oxidation, and this quantifies to what extent the process discriminates between the two stable isotopes of nitrogen. The second part of nitrification is the oxidation of nitrite to nitrate, catalyzed by nitrate oxidizing bacteria, NOB. The marker gene often used to study that is NXR for nitrite oxidoreductase, and it has this unusual inverse isotopic enrichment effect of about -13 per mil. And if you have this classical nitrification under certain circumstances, like the ones encountered by von Brandt and colleagues in very early on, 1939, you can get classical cascades where you get organic nitrogen remineralized to ammonia, and then ammonia oxidizers growing on that, producing nitrite, and then nitrite oxidizers growing on that and producing nitrate as a sequence. This relies on these two steps being catalyzed by two different microbes. And recently, however, an additional way of doing nitrification has been found in 2015, where they found this bacterium called Nitrospira inopinnata that can do the entire process of nitrification all the way from ammonia to nitrate within the one single organism. So this would be up here would be the case that we've seen up there. And this is the new case where it's done in a single organism. In my research, I use a very wide range of oceanographic methods that I won't introduce in detail, but I want you to encourage to ask about them if you're interested in any specific methods that I showed results of during this talk. I just list here sort of the range that goes from oceanographic measurements to stable isotope measurements, chemical concentration measurements, numerical modeling, and a bunch of molecular methods to identify the taxonomy and functional potential of the microbial communities in the environment. These methods I applied to two different field sites, Pow Lake in British Columbia and the Bedford Basin right next here to Halifax. Bedford Basin is 70-meter deep, eutrophic, anthropogenically impacted coastal basin, where the BIO has been running their weekly time series since 1992. And due to the seasonal stratification that causes a lot of seasonal and sub-seasonal dynamics, it's a really interesting place for a time series. And we did weekly sampling there between 2014 and 2017. By contrast, Pow Lake is a much deeper, 350-meter deep former fjord. It's now a lake that got disconnected from the ocean at the end of the last ice age by isostatic continental uplift that raised the sill of the former fjord above sea level, isolating the water body that's now in the lake from the ocean, and also isolating some of that seawater, trapping it inside what's now the lake. So we find this ancient seawater at the bottom of the lake, which gives the lake a permanent and stable stratification. And due to this stability, the time series is not too interesting there. So we did vertical profiling on 2 occasions. And I'll start with the results from Pow Lake, and I extend sort of what's at the core of my thesis about Pow Lake, titling it Toward an Understanding of Nitrogen Fixation and Nitrification in the Lake. So starting from this thesis chapter, there was a publication published in 2019, together with Duani Desai and Julie Laroche from the biology department and Rich Pavlovich from the University of British Columbia, which was the first biogeochemical characterization of the water column of Pow Lake. And I'm showing geochemical profiles from that study, where here we see 350-meter deep water column of Pow Lake, and in the blue dashed line, we see salinity going up to 17 grams per kilogram in the bottom water of the lake, indicating the presence of this ancient seawater that got trapped at the last ice age when the fjord became a lake. And due to the salinity, we get the density stratification that in turn causes oxygen to become undetectable below 130 meters because of insufficient replenishment with oxygenated water. And below this oxic-anoxic interface, we see this typical sequential accumulation of reduced dissolved chemicals like hydrogen sulfide and reduced metals that we also see in marine sediments. Due to the same reason, we see it in sediments due to anaerobic respiration processes in the absence of oxygen, like sulfate reduction, iron reduction, and so on. In terms of the nitrogen cycle, we see here in red the ammonium concentrations being very high in the bottom water. And you can appreciate how it basically, from this maximum at the bottom, it diffuses upward toward the oxic-anoxic interface. And where oxygen becomes available above the oxic-anoxic interface, we see, as expected, a nitrate peak, indicating nitrification, which requires oxygen and uses the ammonium diffusing up, like basically where the upward diffusing ammonium overlaps with the downward diffusing oxygen. Nitrification can occur to create this nitrate peak. And I want to point out this very high ammonium concentration once more, because I'll be talking about nitrogen fixation in the lake in a moment, and it's it's not necessarily expected to find nitrogen fixation under high ammonium concentrations, given that simply assimilating ammonium is less energy intensive than nitrogen fixation. And so that's an enigma that is interesting to study in Pow Lake. One more time back to this 2019 paper. Part of what we did there was to look at 10 metagenomes from 10 different depths, focused around the anoxic part of the lake, studying actually slightly above the anoxic interface, and non-linearly spaced metagenomes from different depths down to the bottom of the lake. And metagenomes are basically, you take a sample from the lake water and sequence the entire DNA, and you get ideally all the genes from this batch of water. And from this big mass of genes, we extracted again functional marker genes of interest from the carbon, nitrogen, and sulfur cycles that could give us the changing functional potential of different processes over depth. And just as some examples here, this red one would be functional marker genes for aerobic respiration. This purple one here would be for nitrite oxidation. And so as expected, we find functional potential, genetic potential for nitrogen oxidation highest, where there's still some oxygen just at or above the oxic-anoxic interface, and then less of it as we go down into the anoxic zone. The next thing we did with these metagenomes was to reassemble them back into genomes using metagenomic binning techniques that create so-called MAGs, metagenome assembled genomes, which are basically draft genomes where you try to make sense of this big pool of genes you get from the metagenomes and try to reassemble the genomes of microbes from which these genes stem. And this allows us to link the functional information from metagenomes back to the taxonomic identity of the microbes that possess these genes. And one takeaway from this 2019 study was that some of those features we detected from these DNA analyses were unexpected and can set the foundation for further study. And that's where I want to pick up on for the rest of my talk about Pow Lake, where we found two of these MAGs associated or having containing the NIF genes, the functional marker gene for nitrogen fixation, identifying them as potential diazotrophs, which means nitrogen fixing organism. And one of those diazotroph MAGs was affiliated with the gamma proteobacteria. It was highly abundant at the oxic-anoxic interface. It was probably a crenothrix-like gamma proteobacterium. Crenothrix are highly abundant methanotrophs at oxic-anoxic interfaces in stratified lakes. And we also found 16S RNA gene sequences highly abundant oxic-anoxic interface. So probably this MAG represents a crenothrix-like gamma proteobacterium. This MAG is also fairly complete, 97% complete draft genome. And in addition to the potential for nitrogen fixation, it has the potential for methanotrophy, so methane oxidation, as well as denitrification from nitrate to N2O. And the second MAG of a potential diazotroph we found was associated with the delta proteobacteria. It occurred further down in the water column where sulfite was available. It was less complete than this gamma proteobacterial MAG. And it's probably, so based on other genes inside that draft genome, it's probably a sulfur cycler and possibly a nitrogen reducer. So all of this is based on DNA, which always has this caveat of DNA doesn't necessarily equal activity. So they could have the genes and not express them. So you need kind of different lines of evidence to establish more firmly that nitrogen fixation actually is active in the lake. And one of those ways is to do biogeochemical experiments where you add 15N labeled N2 to batches of lake water and incubate them. And then basically after incubation, if there's active nitrogen fixation, you would expect the biomass that you filter out of this water to be more enriched in the 15N, having taken up the 15N and 2. And here we see in blue the natural abundance, so prior to incubation with 15N, and in red after the incubation with 15N. And we see that in some cases at the oxygen-oxygen interface and in the bottom water, there seems to be an indication that 15N2 has been taken up during these incubations. And a different line of evidence for active nitrogen fixation comes from finding NIFH RNA, so transcribed DNA, which gives them found in samples between 120 and 180 meters, and RNA, unlike DNA, gives you better evidence that for actually active nitrogen fixation. Still, it's fairly early days on this, so next steps would be to analyze the taxonomy of these NIFH genes, which might enable us to link them back to these MAGs if this RNA is found that is associated with the DNA from these MAGs. And the second one would be to try to replicate these incubations, since we don't have these 15N incubations, which we haven't done very many of so far. A second line of research that came out of this 2019 paper on Pow Lake was to look at nitrification. And part of this is that we found a MAG that is probably represents a Chromamox bacterium. So to remind you, Chromamox is this newly discovered type of nitrification where one organism, Nitrospira or Inopinata, can do the entire oxidation from ammonia to nitrate. And in Pow Lake, we found this MAG that contained both the marker genes for ammonia oxidation and nitrite oxidation. And it had its highest relative abundance in metagenomes from the nitrate maximum depth, so around here, where I said before, the upward diffusing ammonium is being used as soon as oxygen becomes available. So right here, we find this MAG most abundant, or genes associated with this MAG. And also, if we look at our 16S RNA gene sequences, we find Nitrospirus here, so the taxon this Chromamox bacterium is from, most abundant around this depth. So this seems to add up, but it's even earlier days on this one. And next steps will be to analyze the molecular data further, also in the context of stable isotope data we have on the delta 15N of ammonium, nitrate and nitrate, which unfortunately today I wasn't ready to show any of, but this will be interesting to explore the effect of Chromamox on nitrogen isotope biogeochemistry, which hasn't been done too much, if at all, given how newly discovered Chromamox is. And also a similar story about studying Chromamox in a stratified lake, where I'm not sure it has been studied before. With this, I'd like to switch to the Bedford Basin, so the research done on the Bedford Basin, and introduce you to the circulation of the basin, which shows stable stratification in summer and fall, when the bottom water is isolated from the two-layer circulation we see near the surface due to this relatively shallow sill. And you can imagine if the water is stratified, the bottom water would be isolated from that top layer circulation. During winter, however, and spring, we get full water column mixing, where the entire water column gets mixed and the bottom water gets in touch with this surface circulation. Also during stratified periods, we can get interruptions of stratification by the intrusion of dense shelf water from the Scotian shelf, spilling dense shelf water into the basin. And in short-lived events, replace the bottom water to some extent. And these events occur irregularly, usually once or twice a year. Sometimes, however, there's years without them. And this is kind of how this plays out over this study time frame, where we did these weekly measurements between 2014 and 2017 in the Bedford Basin. On the y-axis, you see depth in the center of the basin. And if you look at the oxygen concentration in the top panel, you see how oxygen decreases during the summer of each year to minima in the fall and winter due to the uptake of, due to aerobic respiration, taking up oxygen, and insufficient replenishment with oxygenated water due to the stratification. And kind of the opposite picture for nitrate. Nitrate accumulates each year in the fall and winter, indicating nitrification occurring in the bottom water. And that's what we're interested in here. And this is presenting results from a paper that is under review called Physical Mixing Controls and Decouples Nitrification via Biomass Dilution. And it's come out of a very fruitful collaboration with 2 other PhD students from Dalhousie, Brent Robichaud from Julie Laroche's lab in the biology department, who developed the qPCR assays for this study, and Subhadeep Rakshid, who from Chris Alger's lab here in the Oceanography department, who developed the numerical model that I'm about, or that I'll present later on. And so at the basis of this study was the question why we see interannual differences in nitrate and nitrite accumulation. So here we look, from now on, we're going to look at data from the bottom water from 60 meters in Bedford Basin, and we see concentrations of ammonium, nitrite, and nitrate concentration here over weekly data over those 4 years. And if we first look at the blue ammonium concentrations, we see that ammonium, the substrate of nitrification, is plentifully available throughout each stratified period. So no problem there. And then if you look at the yellow nitrate concentration, we see again these maxima in fall and winter. And what we can see quite nicely here is that at the end of winter and spring, the nitrate decreases and evidently gets diluted out of the bottom water by the winter mixing occurring each year in the late fall and late winter and spring. And the interannual differences I was alluding to occur after that. So if you look here in 2016, the winter mixing stops here, and we get immediate increase of nitrate concentration after that. And then there's one of those intrusion events, and nitrate increases again straight after that. In 2015, we get this very long period of winter mixing that doesn't stop until May. And then it takes several weeks, maybe up to 2 months, until we see significant increase of nitrate and nitrite. So to explore this, the molecular data was instrumental. And here I want to focus on the green scatter, which represents the MOA marker gene for ammonia oxidation from archaea, so archaeal type ammonia oxidation. And we see that it has, first of all, it has very similar distribution as nitrate concentration. We see maxima in fall and winter. And then we also see a strong decrease associated with winter mixing, especially here in 2015. And interestingly, we also see the same interannual differences where in 2015, as to nitrate, where in 2015 MOA doesn't increase for a very long time following the winter mixing. And in 2016, we see an immediate increase in MOA here, the intrusion event, and then increase again afterwards. And together with physical data indicating that winter mixing was probably more intense and longer in 2015 than in 2016, this led us to the conclusion that slow population regrowth after strong dilution by the strong winter mixing in 2015 caused these interannual differences in nitrite and nitrate accumulation. And I've talked about nitrate mostly here. I'll get into nitrate later on as well, but just to flag this, that we see these intermittent transient accumulations of nitrite in 2015 and 2017, but not in 2016 and 2014. And all of this can be demonstrated pretty nicely with the numerical model we developed here. And so I'm going to introduce that here, where we have this bottom water box in the model in which these 3 processes of organic nitrogen remineralization to ammonium, ammonia oxidation, and nitrite oxidation occur. And ammonia oxidation and nitrite oxidation aren't only, these processes aren't only modeled as functions of their respective substrates, ammonium and nitrite, but also as functions of the functional marker genes they're associated with MOA or NXR. So the more MOA there's in the model at a given time step in the model, the faster the ammonia oxidation can occur, and the same for nitrite oxidation. And then vice versa, also so basically these these marker genes proxy the functional biomass of ammonia oxidizers and nitrite oxidizers, and this functional biomass in the model can grow if these processes occur. And one more aspect of this model was the surface boundary, which isn't modeled, but it is fed with empirical data from the near surface water time series of oxygen, ammonium nitrite, and nitrate concentrations. And this surface boundary can exchange its contents with the bottom water box through mixing, which is very low during stratified periods and high during winter mixing, and can be modified depending on how strong winter mixing is. And that was done based on oxygen concentration changes in the bottom water. And this is results from the model where the line plots are output from the model, and scatter plots are empirical data of the same things, the same parameters. And in the top panel, we see oxygen concentrations as well as the modeled mixing. I want us to focus though on the middle panel here, where we see again this in the scatter, we see the data we saw before as stacked bar charts of ammonium nitrite and nitrate concentrations. And now we see the model as well, which can resolve or fit these empirical observations fairly nicely over the entire 4 years. And we made a point here to model the entire 4 years with a single set of rate constants. So that's not a variable that changes in the model. And yet the model can catch both this delay in nitrate accumulation in 2015 and the immediate nitrate accumulation in 2016. Actually overestimates it a little bit. I'll get back to that. And it's a similar thing with the functional marker genes for ammonia and nitrite oxidation. It can get this delay in their regrowth in 2015, as well as the immediate regrowth in 2016. And this is even more nicely demonstrated by this little model experiment we did, where in the year 2015, the one with the delay in nitrate accumulation, we modified the intensity of winter mixing up until March. So here the straight lines are the model we saw before, the sort of standard model. And then in the dashed lines, we see a mixing increased prior to March 2015. And in the more dotted lines, we see mixing decreased prior to March 2015. And in the model that decreases mixing, we see less of a dilution of the functional microbes, of the nitrifiers, of their genes, and they can regrow faster. And if we mix more intensely, they regrow later in the year. And this translates into the nitrate concentrations here, where if we mix more intensely before March, then nitrate accumulation is delayed even further, and vice versa if we decrease the mixing. And possibly even at least similarly interesting, we see in the nitrite. So we have these nitrite peaks I alluded to, and the model can catch that fairly well in the standard model. And if we increase the mixing, it actually pronounces this nitrite peak further. It also delays it in the year. And that's because if we look back down here, so the ammonia oxidized, they did this gap here between the ammonia oxidizer regrowth and the nitrate oxidizer regrowth grows as we increase the mixing. And that's when nitrate can accumulate, because the ammonia oxidizers produce nitrite and the nitrate oxidizers haven't grown their population enough to get rid of it. And so conclusions from this is that mixing can control nitrifier biomass, timing of nitrate accumulation, and timing and degree of nitrite accumulation. And to this last point, there's actually a little more that I want to talk about, how the nitrite accumulates. So for this, I go back to this plot we looked at in the beginning of the Bedford Basin discussion, and add this plot which shows bacterial ammonia oxidizers in stacked bars. And these are just different types of bacterial ammonia oxidizers. So just focus on the sum of the stacked bars and compare them to the nitrite concentrations in this red scatter, which where you can see that every time, both times we get this significant accumulation of nitrite in 2015 and 2017, we also get a peak associated with that of bacterial ammonia oxidizers. And this makes a lot of sense if you know from literature that bacterial ammonia oxidizers are faster growing than archaeal ammonia oxidizers. So what we think happens here is after the strong mixing in 2015, the bacterial ammonia oxidizers can outcompete the archaeal ammonia oxidizers, which are represented by these green. This MOA is the archaeal MOA, and you see how this the bacterial ammonia oxidizers peak prior to that. So they outcompete. They all get diluted by the strong mixing 2015, and then the bacterial ammonia oxidizers grow back faster. And because they grow faster, they don't only outcompete the ammonia oxidizing archaea, they also produce nitrite pretty fast and overwhelm sort of the nitrite oxidizer community, which also has to regrow. And only when they regrow, as the purple guys up here, they can get rid of the nitrite and resolve this transient nitrite accumulation again. So the takeaway from this is that the fast ammonia oxidation kinetics of bacterial ammonia oxidizers enhance nitrite accumulation observed in 2015 and 2017. And the overall implication of this study is that we show that mixing can affect the nitrifier biomass and community, and due to that, the ammonium nitrite nitrate ratios that we encounter at given points in time. And this in turn, if you remember from the beginning of my talk, can affect phytoplankton ecology, at least in theory, since we know from literature that the ratio of especially ammonium to nitrate can affect the phytoplankton community composition. And then this can be extended on that far side, given that mixing is atmospherically driven. And we think that winter temperatures have a lot to do with mixing in Bedford Basin specifically. And so a climate and weather affect mixing and lead to this causal chain, potentially all the way to phytoplankton ecology. One sort of outlook item is so one potential thing to move forward in Bedford Basin research on this account is to look further into this competition between archaeal and bacterial ammonia oxidizers. So here I reproduced the plot again that we looked at before with the model output. And down here is the model runs slightly differently with a different rate constant for ammonia oxidation to better fit the increase in the marker genes, in the MOA marker gene, which we overestimate in the standard model here. But when we make the rate constant for ammonia oxidation sort of smaller, slower, we can get this much better. And this is sort of what could be seen basically as a prototype for a more complex model that could be created in the future, where there's two types of ammonia oxidizers, two kinetically distinct types of ammonia oxidizers, one like an archaeal type and one with slower ammonia oxidation, and one bacterial type with faster ammonia oxidation. I hope I'm doing okay on time, and if I do, I will introduce a little bit one more aspect of the Bedford Basin research we've been doing, which is adding to this data I've been presenting before, the isotopic signatures of ammonium nitrite and nitrate. And again, this work was in collaboration with Subhadeep Rakshid and Chris Alga's lab. And we used the same model as I introduced before and added to it isotopic fractionation factors that for ammonia oxidation and nitrite oxidation. Otherwise, the model was had pretty much the same. And for this study, we focused on 2 of the 4 years, 2014 and 2017, partly because they represented very nice natural experiments. Where in 2014 we had no significant role for bacterial ammonia oxidizers and no nitrite accumulation, or no significant one. And in 2017, we had an important role for bacterial ammonia oxidation and large nitrite accumulations up to 8 micromolar. And I won't go into too much detail here, just present again the scatters are empirical data, and the line plots are output from the isotope model. And in this row of the figure, we see ammonium concentrations in black, and the blue is the delta 15N of ammonium. And we see that the model can catch this increase of ammonium associated with nitrification late in the fall very nicely. The model has some trouble with the nitrite isotopes. That's partly because of these small scale changes that aren't captured by the model in nitrite concentration, which you actually can't see here, unfortunately, sorry. The model does capture, again, nicely the isotopic composition changes of nitrate, so delta 15N of nitrate, catching these minima that we see every year in the fall associated with nitrification, and then the increase following these minima. And at the core of the study was to look at these periods of intense nitrification in fall, which happen to be, as we think we can demonstrate in the paper, closed systems in the sense that very little ammonium and organic nitrogen, so potential substrates of nitrification, are added at this point in the year. So and this allows us to define these as a closed system, which in turn allows us to apply these closed system Rayleigh models, which we see here. And these give us isotopic enrichment factors for ammonia oxidation for the 2 years. So during these empirical Rayleigh models give us these isotopic enrichment factors for the 2 years, which aren't significantly different from each other. And then we can also derive these isotopic enrichment factors from the numerical model, which again are not very different, are very similar actually to what we get from the Rayleigh models. And so in conclusion from this study is that we got these isotopic enrichment factors for ammonia oxidation under controlled yet realistic in situ conditions, which I like to call best of both worlds, because usually these enrichment factors are determined in the lab on enrichment cultures of nitrifiers or even pure cultures. And here we can have relatively controlled conditions because of all these measurements and the very highly resolved time series measurements we do in the Bedford Basin in the field. Furthermore, good news for people who do numerical models of stable isotopes of nitrification. So the isotopic enrichment factors determined from the numerical models and the empirical Rayleigh models agree fairly well. And as shown in the previous slide, this detailed description of temporal changes in nitrogen isotope signatures associated with nitrification in the field is fairly unique and will be able to be used in the future as sort of as a blueprint, maybe also for studies that have less resolution in stable isotope measurements to understand details of what's going on in isotopic signature changes associated with nitrification. And this leads me to my general conclusions and outlook, where for Pow Lake, I think that CHROMOMOX and nitrogen fixation are two aspects that can be explored further in this lake. The CHROMOMOX is still quite new process, a newly discovered process, and its environmental distribution and effect on isotopic fractionation is still poorly understood, and Pow Lake provides an environment that can be studied. Then similar for nitrogen fixation, where we have the ability to study nitrogen fixation in the presence of high ammonium concentration, which has been seen before, but it's still an enigma to some extent why nitrogen fixation occurs under high ammonium concentrations. And then for Bedford Basin time series, specifically the bottom water time series, which has long been a little bit of an add-on to the near surface time series, since the time series started as a phytoplankton time series where people were most interested in the near surface water. But the bottom water is actually a really good place to study many aspects of nitrification. So as we showed, mixing there controls nitrification and the nitrifier community, and connects atmospheric forcing and mixing to nitrogen speciation and potentially even to phytoplankton ecology. And it's also a good place to study this competition or succession of archaeal and bacterial ammonia oxidizers based on their physiological and kinetic differences. And for future research on that, more detailed models, as I laid out, mesocosm experiments and rate measurements can be ways forward. More generally, but also affecting the nitrogen cycle and nitrification of the bottom water very much, is the role of sediments, which is still poorly understood, but I know that Subadeep and Chris Alga are working on this. In terms of physical oceanography, I think there's a challenge to try to better characterize the conditions that drive intermixing intensity and duration. So we ran into this in our study where we where it wasn't, so it doesn't seem to be clear exactly which conditions would cause differences in winter mixing intensity between years in Bedford Basin. And that's always surprising with Bedford Basin, where it's been studied so well. But I think this is really one potential area. I hope I don't go beyond my expertise here, to say for physical oceanography to study in the Bedford Basin and in more detail. And more generally, I think these results stress the importance of high resolution in situ measurements in natural laboratories, particularly the importance of well-resolved multidisciplinary time series, and show that they are very useful to study highly complex biogeochemical systems. And with this, I'd like to thank all of these people, first and foremost my PhD advisor, Doug Wallace, as well as my co-authors and committee members, and these funding institutions and academic institutions that have been part of my research, and all of the people who helped me do my research, especially the research groups, the Gaussian group I worked in, and the other research groups I worked with during my time here at Dalhousie. And I thank you for your attention, and I will take questions next.
Great, thanks for the talk, Sebastian. Do we have any questions out there? I think probably the easiest way is just to unmute yourself and jump in. Maybe they asked all the questions they wanted to ask at your defense. It's possible.
There are a lot of questions at the defense.
Yeah. Okay. I don't see anyone jumping up and down. So, well, you know how to reach Sebastian if you do have a question at some point. He's around for at least the next little while. So yeah, thanks a lot. Oh, wait. There's a question from your supervisor, but it's just one word.
Very cryptic one, very cryptic question, Sebastian. You mentioned mesocosms.
Mm-hmm.
So.
Is that the question?
Yeah, that's the question.
Well, so
is Bedford Basin a mesocosm, or it's more a macrocosm, right? So.
Yeah, I mean, we can discuss about that for a long time. I think we like to some extent, we like to think about it as a mesocosm or natural laboratory, but then so the value of doing actual mesocosm experiments with some of the premises explored in the basin is to actually control even more for some of the factors. Like, for example, those intrusions. The intrusion events are interesting in and of itself, but in the research we've been doing, I think they have been mostly in our way. So it would actually be have been like having years without these intrusions would have made it much easier to analyze the data. And so this is one thing that could be excluded by these mesocosm experiments. Yeah, and just generally, I'm sure there's many other things which can be controlled even a little better, and thereby demonstrated more cleanly in mesocosm experiments than in the Bedford Basin, which is still to some extent a mesocosm, I would say.
Okay, anyone else? Well, with that, I'll say, oh, Ruth is jumping in.
I do have a question. That was really interesting. And obviously I'm interested in ocean mixing, but I'm curious to what extent you think that these results from the Bedford Basin would apply in the open ocean, where you don't have, where it's not so shallow and yeah, you don't have such a confined region. Do you think that these results are more generally applicable?
Yeah, so I think it's a bit of a stretch to go all the way to the open ocean, because it's like, I'm assuming you refer to sort of the mixing part of the results and how it affects nitrification, where in the open ocean there's not these conditions that you kind of need. This intermittent mixing and stratification is kind of the condition you need to get these effects, and it's harder to imagine to have situations like that in the open ocean. But certainly in different settings in the coastal ocean, you could like shelf areas where there's intermittent upwelling and systems like that, where you have these intermittent situations between stratification, more stratified periods, and mixing. I think it's applicable beyond the Bedford Basin, but more like in the coastal ocean than the open ocean.
Okay. Any other questions out there? I think that's it for real this time. Okay.
Yep.
Thanks a lot, Sebastian. That was
Thank you, Dave.
really interesting.
Thanks, everybody.